Thank you. Welcome everyone to Stability Hub's Office Hours webinar, where you can submit stability-related questions, request discussion of a topic, or share experiences on the same. A few housekeeping notes for us today. It adds a lot to the event if you can turn on your camera. We can deal with passing children and cats, and it's fine to be having your lunch while muted. Participants should stay muted unless invited by the moderator to unmute, or if you need to raise a clarifying piece of a question. Questions and comments can be submitted using the chat feature. We may also conduct polls through the Zoom raised hand feature shown on your attendance roster. We just ask that when you raise your hand through the Zoom poll, that you be ready to take it down when we say, so that we can go on to the next poll without messing up our count. This meeting is being recorded and will be posted in the articles and videos section at stabilityhub.com. Our October monthly newsletter article related to today's theme is entitled Stability Intelligence May Be Your Smartest Move, and that's available in the site blog section if you care to do more detailed reading on the topic. Our panelists today include Susan Cleary, Director of Product Development at Novatech International. She's our Stability Software SME. Jason Doust, Brand Director and Monitoring Solutions at LLab, speaking to us about chamber monitoring and chamber things today. Emily Dickinson, Associate Director of Quality Assurance with Clinical Americas, Stability Statistics SME. Patrick Kelleher, Director of Physical Sciences at Freethink Technologies, will be covering accelerated stability. Chris Latos, Manager of Stability Services at Hollister, Inc., covering combination products and devices for us. Hugh Linton, Design Engineer at Parameter Generation and Control, addressing stability chambers. Walter Routh, founder of Sagana Consulting and associate editor at Stability Hub, one of our generalists. Ken Victor, U.S. Laboratory Manager at Lighthouse Instruments, covering instrumentation and testing. And yours truly, John O'Neill, founder of Pharmaceutical Stability Discussion Group and editor at Stability Hub. So we have a wealth of knowledgeable folks here ready to take your questions. With that, we'll send our first question to our panel and encourage you to submit your questions via chat at any point during office hours. So looking at the slate is clean at the moment, and we have a number of pre-submitted questions. So I'll go to Emily Dickinson first. Thinking about stability batches and whether they're early or late in the process, or whether they're a supportive batch, should every stability batch be statistically analyzed or trended? Your opinion on that, Emily?
So you said every stability batch or every batch, just for clarification?
Every stability batch, but you're welcome to kind of parse that out and address each if you
Yeah,
like.
absolutely. So every stability batch should be trended. I think one of the most important things that you can get from that data set is you start to learn the tolerance limits of what your product actually does. So it when you start trending all of your stability batches, you can start to generate what I call a control chart. Well, everybody calls it control chart, which will actually help identify subtle process shifts, especially if your process is robust and well used. Using your stability data along with your manufacturing data to help generate that control chart helps you identify any subtle shifts that you may start to see in your process. That way you can get ahead of any issues that may pop up before they become a potential compliance risk or patient risk.
Okay, well, thank you. And if we have any follow-up questions from the audience, that's fine.
Oh,
We do have
yeah.
one. I think you can probably see that, Emily, but I'll read it
Like,
for sake
yeah.
of time points less than 3 months, like 1 week, 2 weeks, or 1 month or 2 months. What is the common practice regarding the testing initiation and completion from the pull date? So I think that's more addressing Windows.
Yes, test windows. So back when I worked in the stability lab, I'm primarily in QA now. But in the stability lab, especially for our product, which was temperature controlled at the refrigerated condition, for those very short timelines, we would freeze the initial samples. So that way you're maintaining the integrity of the status of that sample while you're continuing testing. Depending on the type of analyses you're running, it can you may or may not be able to speed them up. So if you have an assay, for example, that takes 2 days to run and a day to integrate and get results reported, you can't just speed that up because that's the limit of the test itself. So the way that we mitigated that risk and maintain the integrity of that sample time point was to put it in what we called the stasis condition. So if you have a room temperature refrigerated product, if you have data that supports freezing that product, then you would freeze it to maintain the integrity of that time point.
Very good. Make mention back in a couple lifetimes ago, we did the same practice, which is great scientifically. We just got challenged by a regulatory person that wanted to see data, as you mentioned, that it was okay to freeze, that there was no downside to that.
Mhm.
And once we produced that data, all was good. So just caution people, don't do that without some justification, some data in your pocket, just to
Which
make sure you can answer the question.
Yeah, which then just goes on to further make a better or more robust stability profile for your product if you have those other conditions, whether it's frozen or accelerated, like super accelerated, where you're trying to get a degradation profile.
Mhm. Okay, great. Thank you.
You're welcome.
Let's see. Let's go to Chris Latos. Are there any changes or new provisions to combination product stability practices coming, particularly with regard to the new ICH Q1 guidance? But if you know of any others that are out there, what kind of things are you paying attention to these days, Chris?
Yeah, so it was interesting in the Q1 draft, they do actually have a small section. There's not a lot of detail, but there is a small section on combination drug device products. And they mentioned that the functional performance of the device itself is outside the scope of, Q1. Uh, Basically follow same principles as you would for, drug product attributes. So not a lot of detail per se, but at least, there's two or three paragraphs on it. There will be an upcoming general chapter from USP. So this is USP 1157, which will give probably much more detail on drug device combination products, really focusing on the drug attributes as opposed to device attributes. So, yeah, more to come on that.
Okay, thank you. Watching for the audience questions, and I can use another pre-submitted one here for Hugh Linton, our design engineer from Parameter Generation and Control, one of our sponsors here at Stability Hub. Let's see. I want to go to a different one here. It has to do with all the things we need to worry about refrigerants in our chambers that cool things down. Do changes in the refrigerant industry mean anything to us, particularly, I guess, with the focus on the environment these days? What can we use? What's escaping into the environment? Can we specify a future-proof refrigerant for service? So, Hugh, what do you think about that?
Yeah, the bottom line is it's complicated, but I can simplify the complicated a little bit, and that is so some most people don't actually have to worry about what refrigerant is in their machine because, well, the machine works or it doesn't. It's sitting there chugging away. It's doing its thing. Anything that has the ability to cool a chamber is typically going to have a refrigeration system in it, though. So this, it does bear on a lot of things. Um, Historically, we started back with R22, which was a chlorofluorocarbon, an ozone depleter, so we had to get away from that. And then we moved to R404A, and well, the global warming potential of that was too high, they decided. So we had to move to 134A and other refrigerants. Well, and now coming down the pipeline, the build global warming potential of 134A and other blends are too high, which means that during the construction phase of any new stability storage system, they have to be cognizant of what the refrigerants are that are being used in their machine because the refrigerants, might not be available in 5 years, or they might be heinously expensive in 5 years if the machine needs a top up. And, the framework of how their machine works might drastically change over a period of just a couple of years with the time frameworks we're looking at right now. And the big one is that even the blends that are, you know, okay in terms of global warming potential, they usually include 134A, which is being deadlined in general use. So the big change that this means for a lot of people is that technically the refrigerant that's going to be cooling their space is going to be flammable. So all of a sudden you got to worry about are your service techs for the machines certified for handling flammable refrigerants? Do you have an appropriate mechanical room for where the conditioner is located? All these and more issues are things that a professional is going to have to bear in mind as they look to develop a new stability space because it's all changing right now. Uh, to make things even more complicated, the less flammable refrigerants that were developed by the industry for reasons of safety, they're not very exothermic when they burn. Well, the problem with them is if they do burn or they do get outside and break down, um, they're polyfluorinated alkanes and produce chemical compounds that people don't like in the environment. Europe is full throttle on getting rid of the polyfluorinated alkanes completely and pretty much forcing everybody to use CO2 and propane and butane. So it's a wild landscape out there. And, the conditioner that you buy to condition your space today might not be the conditioner you can buy tomorrow just because everything has changed. This even leads to structural changes in the conditioner.
So it sounds like for us chamber users, we need to keep an eye on those kind of things and stay in close contact with our chamber manufacturers to see if they're going to be able to handle anything coming along that might pertain to some of our older chambers and older systems, that kind of thing.
Right. Be aware definitely that you can't necessarily replace like for like in the future. It's not going to necessarily be possible. You might have a stockpile of one refrigerant and a refrigerant tech that's used to working on a machine that's set up a certain way, but the whole physical design of the machine might have changed because it's got to deal with a very low charge limit refrigerant. And, maybe all of a sudden you find that you have to put up a refrigerant detector in your mechanical room. So it gets complicated.
Okay,
And
well,
it's
thank
only
you.
and it's only going to do more so.
Thank you for those calming thoughts you have for us.
Could I ask a
Be
follow-up?
afraid, be very
Uh,
afraid.
okay, follow-up question.
Follow-up on that is, are there technologies that you could share about that are eliminating or could eliminate the coolants? Like, there's a Stirling and heat pumps and things like that. Are any of those coming up that will be more involved in this?
I don't think typically you're going to see a whole lot of that. Mostly you're still going to be dealing with systems that have standard refrigeration cycle. Uh, and for attempting ICH conditions, it's not necessarily going to be the case that you're going to be able to have, say, a swamp cooler on the roof to cool the air because, well, that comes with its own complications. There are other ways that to obtain the necessary cooling to control your space other than refrigeration cycles per se, uh, but each of them is going to come with substantial infrastructure complications to make a reality.
So for the time being, Walter, no erecting windmills over your stability chambers. Let's shift gears for a minute here. Cheryl has submitted a question. In pharmaceutical programs, active ingredient degradation often defines shelf life. For topical OTC products, where actives remain stable, for example, sunscreens, but physical or sensory changes drive consumer rejection, how might we apply similar modeling or statistical approaches to those non-chemical attributes? So let's see, this might touch on Emily's expertise or some of our generalists. When the active ingredient is not the problem, but all the rest of the product goes south on you. What do we want to track and how do we do that? Is there a scientific approach or is it just a matter of recording that it only lasts for so many months and then fails a physical test? Any thoughts, Emily?
I mean, I guess my question back to that would be is one, are your inactive ingredients assessed in any way on stability? So, for example, if you're talking about how like the feel of a product. So is it something that's more generalized when you're looking at a test like appearance or those physical characteristics, or are you talking more the chemical aspects of it? Because I think with the physical characteristics that can be built into the acceptance criteria on stability, as far as the general feel, the appearance, if that is what is truly driving the consumer.
So
Or
things like color, if you had a color chart or something like that, that you could measure and watch it degrade over time.
yeah, because I mean those things aren't necessarily impactful to the actual action of the product in the API, but to Cheryl's point, that could be something that would drive a consumer not to want to use that product past a certain point.
Mhm. Maybe there's a scale out there called going, going, gone that we could apply over the course of several months. But
I mean, I'm one of those people. I wouldn't call myself a hoarder, but if I have a lotion that's one of my favorite scents, and say, for example, Bath and Body Works discontinues it, I'm gonna hold on to the very last drop of that bottle. But sometimes if you look at the bottle itself, it's got that little container with the lid on it. It'll have whether it's 12 months or 24 months. Once you start getting close to that, like the smell's not necessarily there. So that's the consumer piece that I think Cheryl is talking about.
okay, well, thank you. Any other input?
I have a little experience with that. I mean, so there's some other attributes for those types of products. I mean, viscosity might be important, so you can measure viscosity as a function of time. pH might be important. Also, if there's preservatives, you probably want to do the antimicrobial effectiveness test, like USP 51, which would be a pass-fail. Wouldn't be something you'd necessarily trend, but yeah, those are some of the things that we've looked at before.
So we can't apply statistical models necessarily, but we can come up with a scale or a predictive meme that would say that generally by so many months, this is not going to work. And here's why. We can see it going through these stages on its way there. And then maybe put a label on what is acceptable to Emily's point. Not so good, but still able to use kind of thing, or would not put it out for company to use. Might be some of those criteria that we go through. I know I did a whole lot of over-the-counter products during my career, and we went through all kinds of measures. One of the things I worked on was camphor gel, and we could not get it to stop oozing oil out of the tubes that would come out the bottom. And they finally solved the problem by creating a package that looked camouflaged so that you couldn't tell there was oil leak inside the tube. Yet the product was still good. It was just not pleasing to look at and oil escaping. So I think we have some creative ways that we can deal with these things, but it's unfortunately is probably wouldn't fit too many predictive models.
No, that I would actually push back on that, John. And I can say from experience at Freethink, we have occasionally done accelerated stability testing, building models, predictive models for some of these characteristics. Color change, absolutely. Viscosity, absolutely. Loss of preservative, absolutely. There are ones that it doesn't work. It's harder for things like phase separation or precipitation. Those can be non-Arrhenius behavior and harder to accelerate and model predictively. But the other ones certainly, and it really comes down to a couple of key things. You need to have um, a sufficiently precise measurement technique, and you need to be able to set a failure point. So for color, there are color charts are used, but there are things like Hunter Labs for that. You could buy a colorimeter that quantitates color, and these are used somewhat in the industry. We've published on this where we did a case study where a tablet is changing color on aging. It's still perfectly fine on related substances and assay and all that, but a blue tablet turns green, and now you're going to have patient compliance issues. So that is possible to model and look at the temperature and humidity dependence of the color change quantitatively. Same with viscosity. Brookfield viscometers you can use to measure change in viscosity with temperature, humidity, and time. Yeah, I think I said loss of preservative. As long as you can measure it, that one's usually easier because it's an HPLC method. You could watch as the preservative goes away. You just have to be able to correlate that to at what level will you start failing your antimicrobial testing, as Chris was mentioning. But we've definitely done all three of those and probably others. Yeah, it gets harder when it's things like taste panels or things like that. It's very hard to get it. You need to be able to quantify it reasonably well.
Well, thank you, Patrick. I'm guessing if anything along those lines was going to happen in advancements, it'd be in your laboratory. So we appreciate that. Ken Victor from Lighthouse Instruments, you had your hand up as well.
Yeah, kind of following up on that, I think probably a lot of these characteristics, physical sensory characteristics, are going to be related to essentially the water activity associated with the product. So changes in that certainly have significant impact on like lyophilized products. It changes both the sort of the microstate or the microstructure of the product itself. So I think that's another thing that could be monitored over time to evaluate the quality of the product, and you could easily correlate that with some of these observations about like, oh, it changed its visible appearance or the smell has changed. I think there's a lot of opportunity with that as well.
Thank you. Getting some audience questions in again now, so let's go to some of those. In fact, they're jumping up faster than I can scroll up to see where they are. Let's see, John Stewart, you had a question, and let me get back up to it. When mapping chambers, what tolerance of monitors are being used for both temperature and humidity? And if a device with a wider tolerance is used, for example, a temp tail that is plus or minus 3% RH, is the acceptance range being narrowed to accommodate the inaccuracy? Jason, this sounds like we could start with you.
Yeah, this is definitely up my alley. And the way we typically walk this down is through the test accuracy ratio. And a very common test accuracy ratio is 4 to 1. So if you take, for example, the common tolerances for a stability chamber, which would be plus or minus 2 degrees Celsius or plus or minus 5% humidity, you would take those and divide them by 4. So that 2 becomes 0.5 and that 5% becomes 1.25. That'd be your typical minimum requirement for the loggers that you would want to use to perform mapping on these environments. And that is to help mitigate your potential risk for kind of like you're asking in part 2 of your question, where the logger's accuracy really brings up questions and potential risk that the real environmental condition is actually outside of acceptable conditions, even though the logger measuring it might show that it is within acceptable conditions. So the tighter that you can get the accuracy of the loggers you're using to perform the mapping, the better confidence you have in reducing your risk of performing that profile. So for example, many loggers that are often used for these types of mappings will have a temperature accuracy of 0.1 degrees Celsius, significantly better than the 0.5 that is required by the basic 4 to 1 test accuracy ratio. And temperature is a little bit easier to become more accurate and a more cost-effective basis. So it's not that expensive to get a more accurate temperature reading. Humidity is a bit different though, because humidity becomes increasingly difficult to get more accurate. So most chambers are mapped with loggers with a 2 degree or 2% accuracy. And that's generally accepted within the industry, even though 1.25% would be ideal. It's just so difficult to get humidity sensors to meet that 1.25% accuracy that it's just been generally accepted. So that kind of leads to your part 2, which is you do have to make considerations, and a bit of this has to do with what is the value you're reading versus how close you are to the tolerance. So if your tolerance is 3% and you're right in the middle, and you have your plus or minus 5, you have buffer on both sides. But if your reading is getting closer to your limits, either up or down, you now have to start making considerations. Is my reading actually within the tolerance requirements? Am I within the uniformity requirements of that chamber? And sometimes people will put extra buffer in and narrow down that tolerance to accommodate for the loggers that they're using. But that's challenging. Trying to keep a stability chamber within that ±5 is tough enough, let alone trying to get it to, if you're trying to say ±3 or ±2, like especially with minor temperature fluctuations, that humidity reading will change drastically. So it's always better to use the appropriate equipment for the tolerances that are required. There is another thing you could do that would help with some of these less accurate instruments, and it has to do with doing pre and post verifications. So some of the challenges with some of these devices and their tolerances, the tolerance is based on a wider range of use, but you can do verifications around a tighter range of use. It might find that it actually is more accurate within those specific ranges, or you might actually be able to apply calibration offsets within those tighter ranges and actually tighten up that traditionally less accurate device to make it more appropriate for what you're doing. But that gets a little bit trickier with doing the pre and the post and making sure all that is documented appropriately.
Yeah. Jason, what's the basis for the 4-to-1 ratio? Is that just an industry rule of thumb?
It is. It is
And
just a rule of thumb out there.
yeah. And my specific question, I was auditing a vendor recently and it looked like the chamber was drifting, but it
Yeah.
really was the inaccuracy of the devices that they were using to monitor.
Yeah.
And how do you convince a contract lab to go to higher accuracy? And it's an uphill battle.
Yeah, and usually you have to reflect on the value of what they're doing, the risk if the what they're doing and the measurements that they're taking are incorrect, and you end up having to go back and revisit everything. And it really it has to be backwards driven in terms of the value of what they're performing. Otherwise, you will find people try to use very inexpensive equipment to get away with performing mappings in an inappropriate way. And it's just kind of that penny wise, pound foolish scenario where it comes back to bite them in the long run. And you do have challenges too, where some of those devices do have tolerances, especially the lower quality device, that it itself will drift. And that's something that's challenging to check unless you're really on top of those pre and post verifications to know, really understand the characteristics of that measuring device.
Thank you.
That's a great question.
Okay, Piero De Filippis joining us all the way from the lovely nation of Italy. Hopefully it's nice and warm over there. Says, what is the current FDA expectation for the stability study for commitment batches for generic drug products after primary registration study was performed on pilot scale batches? Is it also expected to perform a study with the same protocol on 3 commercial scale batches? Wasn't able to find reference in the FDA documents that spell this out. So this is more of a generalist question, especially for those of you that are dealing with generics. So I'll throw that open to the panel to see if anybody wants to pop up and hop in on that one. Don't all jump up at once.
Yeah, John, I can comment from previous life. And something to remember is until you have stability data on commercial batches, your dating is tentative. And so the answer is yes, you need to conduct stability on and for generics at multiple conditions on the first 3 batches. And I believe that's also included in the draft ICH guidance. And I'll look for that and put it in the chat if I find it.
Yeah, I noticed over the years when I went to look for things about over-the-counters or generics that lots of times I was going to associations for input to gain some extra knowledge in addition to what the scan information was in the guidances. But the more that you can justify with data, the better off you are. And I think at some point in time, you're going to run into some people who might be asking more than whatever the guidances have asked for. And it's always nice to have some data in your back pocket to go forward with that. So not too terribly a solid answer for me on that, but does anybody else in our audience want to comment dealing with OTCs and their experience with regard to this question? I can't see quite everyone all at once on the screen to see if there's a hand up, but it doesn't look like any at the moment. So Piero, looks like you're kind of stuck with justifying what you do and having a great defense for it. Okay, one from Joey is if more than 3 primary batches have been conducted and ongoing, do we need to put additional clinical supply batches produced after the primary batches on stability program before the MAA, NDA, and BLA? Assumed no or no statistical process changes. So some of you dealing with clinical batches, my experience has been you get the primary batches on, it's going to go in for your registration. You would probably have some batches on stability just to be cautious, not necessarily planning on submitting the data, but having that in your back pocket, and then somebody else can come along and tell me that I'm outdated on that. But I would ask if you have any other input with regarding to those clinical batch studies going on stability.
As far as clinical batches go, depending on what phase you're um, in my experience, we've put every clinical batch on stability because sometimes there could be subtle process changes before you go into your marketing campaign or your registration campaign. So, and also depending on how many clinical batches you're making. So if you have um, low clinical demand, you may only be making 1 or 2 batches a year, depending on the need in the clinic. So putting all those batches on stability to get a good profile of what you can expect going into registration is a good idea.
Okay, yeah, if something goes south on in your clinical trials, the first place they're going to come looking is to see if there is any stability information that might give a clue to what was happening there.
Yeah, also you're going to use those stability batches to help justify the shelf life of the product that's sitting in the clinic. So a lot of the studies that I actually work on right now and supporting, I see expiry extensions coming across my desk pretty frequently. So you're using that data to help justify extension of your clinical product so you can get through the clinical trial without having to make a whole bunch of batches if you don't need to.
Great input. Thank you. Next question. This is more of a regulatory question, but does anyone have experience with ABI that has an OOS stability result? For FDA regulated product, we would of course file a field alert, but for all others, the guidance is not at all clear. The OOS turned out to be a lab error, but we didn't figure out that within 3 days. It's an animal health product if that makes a difference. I've not seen that particular instance pop up, but I imagine it'd be the same for all of us in all of our different products that we have to do the explaining and you can't duck the field alert because it takes a while to get there. But speaks to how fast we need to start addressing it. It's like all hands on deck and no hands go home until you come up with something to see if you can avoid the field alert. Anyone else commenting? Okay, it looks like we stop with that for that kind of information, but certainly we can watch that going forward to see if anybody else pops up. We'd love to see these kind of questions posted in the discussion section on Stability Hub to see if a wider audience might see it and pick up on it with some experience. Syed says, do the new regulations introduce any new requirements for documenting or trending degradation pathways identified during forced degradation studies? We had a panelist dedicated to that very same kind of question, but we lost him due to scheduling conflict today. New requirements for documenting trending degradation. I have not seen any myself. Chris Latos or Walter, have you spotted anything in the more recent documents?
I've read it a couple of times, and I don't remember anything specifically talking about trending, forced degradation. I mean, you're doing your forced degradation to identify your degradation pathway as well as like what impurities could be, generated and so forth. But yeah, I don't think there's a requirement. I don't recall reading that in that document.
Okay.
Yeah.
This is a service that Stability Hub tries to keep up on, watching all the new guidances and regulations coming out, but we're few in number, and so trying to stay on top of that is a major hassle. But we try to list any new guidances, and at some point in the future, it'd be nice if some of us could do some analysis of the new things and comment on this in advance. And we're a group of volunteers, so if anybody has any ambitions to watch new regulations, whether it's across the board or just within one region or country, and alert us to something new, we'd be glad to have you serve in that capacity. It would be a great service to the stability community.
John, I recall reading in the SHQ1 draft guidance, that'll be out in a year or so, that there were some language in there that could be interpreted to mean that to use predictive stability models that you need mechanistic understanding or to know what your degradants are. I remember this because we submitted comments pushing back on that it shouldn't be required that you know the exact reaction taking place as long as you show that your predictive model, as it were, is accurately capturing the rate of degradation. So where the language lands in the end, we won't know for a bit on ICHQ1, but it was a little bit fuzzy, I thought, about whether or not you need to, how strictly you need to identify what the degradant is and what the degradation mechanism is, depending on the phase.
So watching that is a key piece of all our organizations, whether or not you rely on somebody else or within your own house to do that. It's kind of nice if that's an extra job duty of someone who's routinely doing some kind of stability problem and then also keeps an eye on the regulations and relays them to their group. Always a great intelligence factor. Next question, for LVP products, is leachable study required during stability on 3 pilot batches, or is it enough to be on 1 production batch at the end of shelf life? So this may be open to some interpretation, but I've seen a lot of demand for early on leachable studies, which is nice, but by the same token, as this question implies, there could be changes near the end that would introduce new things, and you're going to either be lacking or have to repeat it. So it's kind of nice if you could do something progressive and be systematic about the types of materials that are involved. And then at the end, close to the end, run it on some real life situations to show that, yes, your predictions came true, or you didn't get to the point where you had to worry about that level of leachables coming through. Anyone else want to comment? So I'd say, yes, this is a definite thing about making sure you have some at the end, but not a problem to do some reasonable research early on, as long as you just don't do it early and then call it quits at that and say you're covered for the rest of the life of the studies.
If I can comment, Piero speaking. Can you hear me?
I can. Go right ahead.
Yes. According to my knowledge, the trends of leachable can change during stability studies. So, for example, increasing on storage and then decreasing according to degradation and the current leachable from material. So the only result at the end of shelf life, from understanding, is not acceptable. And another point that I will refer is the recent ICH draft guidance on extractable leachable. And on this guidance, there is a clear expectation that selectable and leachable are run in parallel to the stability studies. So I think it should be on a risk assessment basis, but only performing one result end of shelf life, for example, at submission could be not enough to justify specification for the leachable for the product. I don't know if my Italian English was clear, but
I think so, yes. Thank you for that input. Any reaction to that or additional comments? Okay. Next one from Jennifer Tung. When the new ICH update for frozen conditions from -20°C ±5 and -20 or colder, would there be a concern if we continue to use the -20 ±5 freezer? So people with freezers and products that are typical for those, that's where we're looking to get an answer from.
Could we get clarification, perhaps? Like it suggests that the variability that's of ±5 is what's changing. Is it like being changed to ±3°C? I'm just trying to make sure I understood what the change is that's being recommended.
I think I see the variable dropping between one and the other. So Jennifer, if you want to comment and further, you're welcome to. It looks like people should not have a problem keeping the plus or minus factor in there if that was the concern, because you're doing something that goes above and beyond if we're interpreting the question properly. Did you want to comment further?
Hi, yes. So we while we have drug product frozen, we're following ICH with -20 ±5. And so the freezer operating, we qualify it for operating from -20 ±5. But then when it's actually running, it's basically the worst case that we see is they fluctuating, the normal freezer cycle is fluctuating from -21 to -19. And then, but it's like the other freezer is basically at -20 at all times, just has like normal spikes for defrost. But like our QA has concern about if we continue to use the freezer and we're claiming our products should be stored at -20, and if we have a higher freezer temperature to gather data is not within compliance.
Yeah,
So
the new guideline only says -20°C or below. It doesn't give you like a range. So I think your question is if it's, if you could have like -19 or -18, something like that, would that be acceptable, right?
yeah. Mm-hmm.
Yeah,
That's a great nuance to consider.
yeah.
Yeah, losing that
What
tolerance is unfortunate.
what I'm thinking is because we are collecting data in a warmer condition, still frozen, but slightly warmer, and if the data that we've seen already is still meet our specs. So that should be a worst case scenario, right? And then we're still supporting the colder condition.
Yeah, can you say that the average stays well within the plus and minus sides of the target, or does it consistently? If it was consistently lower, I would be concerned about a regulatory person coming in and having a conversation about it. But if you can show that that's the average is at -20 and that or that you run colder, I would think that would kind of take the club out of their hand on that particular topic. But yeah, I agree. It's unfortunate that the plus or minus thing has fallen out.
Yeah. Right. Thank you.
Okay. John Stewart, looks like you had plugged in some information to a prior question about commitment stability batches and pointing out that it's included in section 15.1 of the draft ICH guidance. So that's more helpful information for us. How can we justify a preservative is chemically out of the specified limits, but it passes the preservative effectiveness test? This goes back to an earlier lifetime for me. We had the same situation. I'm trying to remember how we got around it. I believe that we simply stated that we had a target that we put it in there, and it did not have to remain chemically measurable as long as it kept the preservative effectiveness test. And we had to take that before the regulatory authorities and test that out. If anybody has some more recent information that speaks to that question and how they got around that situation or addressed it or came under scrutiny and didn't get accepted by regulatory authorities, we'd be glad to hear about that. Okay, sounds like my input's got to stand. As in all cases, justification, and that's what you're asking about, is justifying the preservative, is that it still passes the preservative effectiveness test, and perhaps your documentation can shift to say that that's the major goal, is the emphasis, and that you are measuring the preservative. It's out of specifications, but it's probably below detectable limits and still having an effect. You can build a case around that. I know it's scary when you run into that situation. You think, now what are we going to do? But if you can document that batch by batch and have several batches backing you up, I think you have a reasonable justification to send that in as far as you're dealing with the problem. Okay, I see no hands up, so we'll go down to Piero's comment on the extractable and leachable ICH guidance I mentioned is the draft of Q3E in section 3.5 and compliance. Gives more details in section 4.4 for leachable. So our questioning person can go to that and get more information there. Also more input from Hugh. Setting the set point lower to ensure that you don't go above -20 does risk a higher load on your freezer equipment. So there's a caution from the manufacturer if you're trying to defend your position that they ask why you didn't go lower. You can always point them to the supplier of the equipment and say not recommended. It's doing what we asked it to do. It's been validated that way, and that's what we're practicing. And then we'd be glad to hear your story after that, whether or not that flew. Comment? Okay, we can await further questions and we'll toss a few of the previous ones in. Let's see, we don't have Sue Cleary sitting there amongst our attenders. Sue, if you're in the audience, if you would speak up, because I do have a question for software, but we'll come back to that a little bit later if you're joining us later on. One of the ones that pops up every year is how should we clean and disinfect stability chambers without endangering samples and chamber operations? So either the manufacturer or some of our generalists, this pops up. Yep. Syed, are you commenting or you're just unmuting? Okay, I think he was just commenting to somebody else. All right, so back into the practice of the chambers, we want to make sure we avoid raising the humidity by sopping down our walls all the time. We also want to prove that we've not contaminated our samples with any high-powered antimicrobial agents. And so it becomes a matter of what can we use, how often can we use it, what gives us the least impact. So I think it comes down to our practices. There are a couple of recommendations out there from some of the microbiologists as to what to use. I'm not really familiar with those off the top of my head, but I've seen them. And then monitoring to make sure that you're not impacting the chamber environment for very long when you do the cleaning. And then some of the programs for frozen goods are a whole different ball of wax where you got to get those samples out in order to do some cleaning around them and clean at the right possible temperature. So that might involve backup chambers along those lines. Any others want to comment? They found satisfactory methods of cleaning their chambers. Some of us go long, long periods of time and nothing happens. And then suddenly, maybe someplace down the hallway, some other group suddenly finds contamination. And soon your boss is on top of you for not having done anything because you never had a problem. So that can happen with us as well. It's good to think about this in advance and be prepared. Even if you don't want to do anything right now, that what situation will you go to if you're forced into it? Okay, we've arrived halfway point for our timing today. So I wanted to say a few things about Stability Hub before we run out of time at the end and we're too rushed. And halfway through our time today, we'd like to take this opportunity to encourage those of you not yet familiar with Stability Hub to visit us at stabilityhub.com, explore the site for features that are most useful for you, and to sign up there for our monthly newsletter, bringing all things stability together to your attention. Please encourage your colleagues to do so as well. And you could do us a great administrative favor right now. We have a poll that will be coming up in just a moment. If you're attending today because you saw our notice through LinkedIn, please raise your virtual Zoom hand, or better yet, respond to this poll so we can get an idea of how effective that route is. It'll take just a second to hit the buttons there. All 2 people have answered.
All right, I put the wrong question up.
Oh, sorry. Yeah, I was looking too much down at the end. This is question number 2, but
There we go.
can you withdraw this one and go back to the other? I guess just stop sharing this particular one.
Let's see.
We will ask this question in just a minute but I'll be describing.
It might be too late now. Let's see what happens. Let's see. Okay, there we go. Let's try that.
All right, let's try this poll. Practice makes perfect. So we spend a lot of time trying to advertise these, make it known to the people subscribing to the newsletter, but we also realize there are a number of other organizations where stability might just be a sidelight, or you're interested in one particular thing. And so we use LinkedIn to make those people aware of what our topic is and how to get here. So it looks like at least 5 of you are using that route today. So we really appreciate that.
Okay. A significant number.
Okay, we appreciate the input. Let me read the next one, disclaimer, before we go to that poll. If you enjoyed the Stability Hub webinars and their give and take style of discussion and sharing of stability information, you may want to put May 17 through 19, 2026 on your calendar for an extended opportunity to network and benchmark with your stability colleagues. So that would be an in-person meeting. And that's the Pharmaceutical Stability Discussion Group meeting in North Carolina. Further details will appear in the events section of the Stability Hub site. We're also seeking companies that might like to host such meetings in the future, bringing the best of the stability world to their campuses. Contact us through stabilityhub.com/contact for more information. So we'll take our poll. If you're thinking that you might want to attend such a meeting, answering this poll will not commit you to having to do it. But if you think you might have a shot at applying for some travel and a meeting for less than $1,000, not a lot less, but some less, we'd like to know who you are and then keep track of you to see how well you're doing in the future. It's quite a risk to start from scratch again. We haven't had an in-person meeting for 5 years, so we need to be very risk wary when we're hosting a meeting to make sure we don't exceed our resources. So knowing how many people intend to try to attend is very helpful to us.
So unfortunately, the poll
doesn't come back, huh?
I had to cancel it because of the other questions. So
okay, well, you can send us an email in that case, or go to Stability Hub and just respond by filling out the form.
Well, we could just ask people to raise their hand if they, for a yes, if they would be able to, if people could raise their hand, that'd be great.
Sure, we can go old tech, especially if we're doing that for other polls. So over to the chat feature and then give Walter time to count those hands. We would love to see people in person, and you have a much greater opportunity for follow-up and questions that occur 2 minutes after the question went by and you still want to get back into it. There's lots of opportunities there. We also take a tour of the host facilities, get to see what's happening with them and an activity. So there's a lot of networking and bonding going on with, as well as bringing questions.
Okay, great. So I see 5 people. Appreciate your votes. 5 people were saying that they would be able to attend in May.
Sounds good. We'll do future polls as well through the newsletter to get a wider audience to count for that. If we can get up in the 20 or higher area, that pretty much guarantees enough economic oomph to actually hold the meeting. So we'll be watching that. But right now we're intending to hold it on that weekend. That's the traditional PSDG meeting weekend, the weekend after Mother's Day in the springtime. So put it on your calendars. And we're thinking also of making that connect into an office hours segment as well during the meeting. Okay, we'll continue to entertain your questions now. Let's see what we have here. Significant changes in assay applicable to pilot batches or during, got a little bit of during shelf life. Significant changes. The assay meaning the assay results or the assay test methods? I'm assuming results here. So I would say yes during pilot batches, but I'm open to some of the other panelists to ask that question. Anyone dealing with pilot batches that wants to comment? The guidance talks about what's significant. So if you're early on in the process, it can be 5%. And if you're later on and have developed some limits, that can be less than 5%. And John Stewart raised your hand.
Yeah, if you're running accelerated your stress condition at 4075 and you do have the significant change, that would invoke testing of your 3065 condition. And that would be regardless if it's a pilot or a any other batch that were at that condition.
Okay, thank you for that. Any others?
Can I ask a related question on that answer?
Sure.
Is it common practice to only test your 30 if you see a failure on the 40? Is that what everyone does?
Yes.
Yeah, 30 versus if that's the only room temperature condition, or do you have 25 and
Or sorry, 38, something midpoint. You keep it on test, but you just don't test it unless you see the fail on the higher. Okay.
Yep, I agree with John. That's when you test that.
Un-
Although some
unless-
companies, yeah, go ahead, John.
Yeah, unless you're also running for in a climactic zone that needs the 3065 as the long-term condition. That would be the exception.
Yeah, and then some companies, I've always been in favor of this, but I've never found much support. Just drop 25 entirely and go with 30 if our product could withstand it. So there's less data points to have to report. But the case is more like John just described it. It's more backup in addition to the 3065. I haven't found there's not a large club in favor of doing it the way I just mentioned. And while we're with you, Cheryl, adding to this question, is there any good way to avoid mold and mildew when the chamber cleaning topic in high temperature, high humidity chambers? An excellent question. I think regular cleaning is is about the only way I know, and using an effective microcidal, bactericidal solution.
Also avoiding any cardboard or
Mhm.
target material.
Yeah, good point. We used to use cardboard almost across the board in the early days of stability, before the plastic bin people came along. And that helps a lot. Was not only with cardboard can you create a big humidity and wet sink in there that'll release stuff, but you can also bring on mold. And with cardboard, you could also get paper lice. A lot of people haven't heard of those, but we've seen them turn up in stability chambers over the years. And getting rid of them is a real challenge.
Hmm.
Crawling around within all your paper and then making sure that they get out of your chambers.
Another possibility is a UV light. I know with PGC, a perimeter does that, has a UV light for the circulating air and water. So just killing that as you go.
Mm-hmm.
And across,
yeah.
Sorry.
Go ahead.
Well, Walter, is that UV light in the ductwork or is it actually in the chamber?
Not in the chamber, it's in the air handler.
Yeah,
Okay.
I can
Thank you.
speak to that. We normally haven't done a UV light in the ducting or in the chamber itself. Typically, the UV light is offered as an option that's in the water spray recirculating system, so that it's toasting the water system with UV light. And a particular adopter of the UV systems of that kind is the paper industry, because surprise, surprise, if you're running a tappy chamber, you tend to have a lot of small fragments of cardboard circulating in the air, ending up in the sump of the machine, and not necessarily settling out, maybe trying to grow some things. Uh, in terms of chamber wall sterility, I know that the panel manufacturers do offer antimicrobial coatings for the interior of the chamber, but most people don't buy those. I would also caution anybody who wants to clean down the interior of their chamber with a strong acid. If you start the machine up again while that's still in the air, that condensing on your coil is going to reduce its lifespan a little bit. Ask me how I know.
Yeah, I kind of suspect somebody's tried that. Yep, we've heard some horror stories about chambers that went south really fast because of an untried method or something that somebody was not familiar with the negative impact of using certain agents. I'm trying to think. Oh, there's been a lot of controversy about whether or not low temperature stations need to have humidity, and typically it used to be 5 degrees. Some companies would control humidity and monitor, and then over time we found that no one was maintaining humidity at 5 degrees because it put too much moisture in the air, and especially when you had cardboard to do that. So then the practice went over to just simply monitoring what the humidity is. Not a bad practice. You can't get hung for that. It's good to know what your humidity is, especially at those low temperature conditions. It doesn't take a lot of moisture to set the relative humidity level up pretty high.
And if I might speak to that one as well, we do see sometimes that we have a take rate for dryer on a 5C room, because we're maintaining the coil temperature above freezing to make sure that we don't have to go through temperature disruptive defrost cycles. And that means that the humidity in the 5C room is dwelling at a pleasant, like 85%. And for some people that doesn't work. So for some people we augment with a desiccant dryer to pull the humidity level down a little bit.
Mm-hmm. When you're desiccating, is there a safe level that you pull down to? Because you want to make sure you don't dry out the product too much. So I imagine there's some kind of setting that you target.
Well, we technically can control. Excuse me. We technically can control a humidity down around those levels, depending partly on what the ambient dew point is. whether we're going to have to pull in air, whether we're going to have to pull in air to the process to be able to raise the humidity back up again, or whether it has to be humidified. But typically, if we're applying the desiccant dryer, we're just using a capacity control on the desiccant dryer. And most people are shooting for a target that's below a certain level, at which point we're basically idling our dryer. We're not trying to actively control it, and the dryer's not going to necessarily just keep chugging away and making it drier and drier and drier and drier. But it depends. It depends. We have had people who want to actively control a below freezing dew point. It's just usually with the 5C, they're looking at an upper threshold of what's allowable, what the upper threshold of where they're starting to have problems.
Okay. Well, thank you for that input. While we're on the topic of water in the chambers and maybe water in our products, because I know that would make a difference whether or not we're dealing with an ampule or a vial or a tablet that just simply has a screw tap on it. I want to turn to Ken Victor and talk a little bit about water activity and that kind of testing, particularly with regard to USP 921 for moisture determination and 922, the use of water activity testing instead. So you can tell us a little bit about the difference between the total moisture content of a product versus the water activity and how that impacts us as stabilitarians.
Yeah, so it's very common for lyophilized products in oral solid dosage that water content is a critical product attribute. And they typically are linking it to shelf life and stability, but it's under growing recognition, certainly within the food industry, which already does basically water activity measurements. But water activity is actually a better metric when trying to evaluate product stability. And the idea is that the total water content of a product, which is what is measured if you do Carl Fisher assays, is basically taking a look at all the water that exists within that product. But what it cannot do is distinguish between water that is tightly bound versus water that is free. They're sort of that's a very crude segregation of the water population that's in the product. But basically, the idea is that the bound water represents water that is critical to the structure of the molecules in the product and is not going to contribute to degradation processes. While the free water typically has more mobility and it is capable of inducing degradation processes. And so the idea is that water activity is basically a thermodynamic property that is essentially a statement about how much free water exists in your product. So the idea then is that the water activity is a better metric of evaluating the risk of your product degrading or is more correlated with your degradation processes because it is a measure of that free water instead of the total water content.
So would you say that knowing the water content or moisture level can be used as a predictive measure for how long our shelf life is going to last, at least from that aspect?
Yeah, so the statement would be it's better to use water activity as a correlation to your product stability than the total water content is, because the total water content includes that bound water that may not be an issue. Like, it's typically not associated with the degradation processes associated with the product. And that's what I was kind of mentioning earlier, like when they were talking about optical appearances of products, like of change. I think a lot of those is going to be associated with the change in the water activity associated with that product over time.
And I'm looking across the row of pictures here on my screen, seeing Patrick and thinking, is that something they also use in the ASAP realm of things to make a prediction or take some measurements with or for?
Yes, absolutely. When we're doing stability modeling using ASAP Prime, the way that we factor in the effect of moisture on stability is explicitly water activity or equilibrium relative humidity. They're the same thing, just divided by 100. We avoid Carl Fisher data where we can. It's kind of an inherently noisy measurement. We've definitely seen data sets where whether it's measured in the winter or the summer, you get dramatically different results, especially for non-hygroscopic material. But you can also convert between these values if you have the moisture sorption isotherm typically measured with a dynamic vapor sorption instrument. So there are ways to kind of translate between KF water, DVS water, and water activity, just mathematically, if you have a few key pieces of information. But yeah, we would love to see if people started monitoring and reporting water activity rather than Carl Fisher water. It's a more accurate measurement as well.
Okay. Well, thank you both. That's very helpful for us in understanding the impact of the water activity for our products. Let's see, any new questions? Not yet. So go back to a pre-submitted one. Thinking about when going commercial, especially, this could be applied earlier in the process, but when we get close to the end, we tend to get a bigger gap here. How should we handle the gap in time between the date of manufacture and the date on stability if it is greater than 30 days or going out even to 6 months when people consider the amount of time it takes to ship our product around if we make an API on one continent and incorporate it into a product on another and then send it to a third country someplace else to be packaged and then maybe even cross the ocean again and sterilize it. That often runs the clock way out. And so we get lots of questions around what should we do in those cases? We're supposed to use the date of manufacturer as a start of shelf life, or we start measuring what goes on from the time you put it in chambers. And typically we come to the conclusion that we should try to accommodate that gap by acknowledging it in our stability study. So we might have a date of manufacture and a date we put on stability, but then basing from the date of manufacture when the expected shelf life would end and enter more tests around that point. So we can say to anyone that questions that, what about that gap in time? And we can say that we did measure the stability that occurred through that. Now, it's difficult to say that the date of manufacture is a date on stability when you don't have some of the key factors of what a stability study is. So maybe it's stored in a warehouse that doesn't have a tight control over temperature and humidity. Maybe there's no security on that because a lot of us define a stability study as being in a secured place, so no tampering can occur with that. And then possibly the accuracy of the monitoring systems in there, if there's a monitoring system at all. And then, of course, you're shipping things back and forth during that period of time. And so there's varying conditions there. We may or may not have been able to monitor really accurately for periods of time. One point in my career, I did see some product that was made in the UK. No, sorry, it was made in Ireland, and simply shipping it to the UK, it went out of specifications. The temperature rose above 30 degrees C. So there's one report that we had to file on that particular transfer, but then they shipped it to California from that point. But in the process, they subcontracted the shipping to yet another person and didn't pass on a whole lot of restrictions. So instead of flying from London to San Francisco, where it was supposed to end up, they flew it from London to Las Vegas. Don't ask me why they chose that. And then offloaded it from the plane and put it in the back of an 18-wheeler and sent it down through some of the hottest parts of America and ended up with over 80 degrees centigrade conditions in the truck where they finally got some monitoring in there. So those kind of things occur when you're shipping things back and forth. So that all enters into the reasons why you want to do some careful considerations, what happens in between the two periods and what kind of testing you want to do. And that's a lot of verbiage around that. Did anyone else want to comment on what they do to account for the time between manufacture and date on stability to make sure they have good justification for their data that they have? Okay, too much storytelling, so we can move on. Let's go back to Ken. Container closure integrity methods regarding headspace analysis. A lot of change over the course of my career in container closure integrity and what methods we're using. For a while, it was all the older traditional methods, and then a lot of new ones came out. People started shifting in different directions. Where do we stand now for the best methods and what's actually being used?
Yeah, so one of the things I want to point out is particularly with for container closure integrity is and touches base with some of what was asked before is like things with like cold storage. Um, Traditionally, I think CCI tests were designed to just test the container when it was essentially at room temperature. So you would potentially store your product at a cold temperature, but if you use a CCI test method, you would take it out, it would begin to warm up, and then you would conduct a CCI test on that. And the critical thing is understanding is that there are such things as temporary defects. So defects, particularly that can occur only while it is in cold storage. So like your elastomer components have essentially a, a glass transition temperature. So at a point where when it goes below that temperature, it no longer is an elastomer, it becomes brittle. And it also has a differential thermal coefficient of expansion than typically the glass container, for example. And so both of those phenomena will end up generating these temporary defects when it's at cold storage. And so that can have an impact. So there are cases where you have a leak when it's in cold storage. Most often this occurs at things that are stored at -80. So cold air will leak inside the headspace of the container. The container is removed from the cold storage and it begins to warm up and it reseals itself. And so now you have cold air trapped in the headspace, and you end up with a situation where you have a dramatic overpressure in the headspace due to that. And so when you insert a syringe needle into your stopper to remove the product, the overpressure is released, and so the plunger in the syringe will pop out, and the product will spray out of the syringe. So these are ideas is that instead of testing or evaluating container closure integrity just while it's at that warm temperature, by analyzing the headspace contents itself, you are able to identify these temporary defects. These temporary defects also can occur within lyo chambers, where the lyo chamber compresses and pushes the stopper into your vial. Then the vial shells have to separate a little bit so you can remove those vials. So there's a common thing known as stopper pop-up. So and in that time period between when it's removed from the lyo chamber and it gets crimp sealed, that vial can have a leak in it. And so you may crimp seal it and then test your CCI and you think, great, I don't have a leak, but you wouldn't have identified that phase in between it was before it was crimp sealed. So again, that's a situation where the headspace contents will change. And if you use headspace analysis, you'd be able to identify that. Whereas like if you're doing traditional testing like a vacuum decay system, you would not be able to identify those temporary defects.
Any specialized considerations for lyophilized samples that would be different, or is that the same?
Yeah, so that's what that second thing is for lyophilized samples is you could have a situation where you may have a criterion is like under my lyophilization process, I want to be sure that I have removed all the oxygen because my product is oxygen sensitive, and you assume like, okay, I'm going to lyophilize it and then I backfill it with half an atmosphere of nitrogen. But if you have that temporary leak, you may ingress oxygen into your vial before it is crimp sealed. So then afterward you say, oh, I've crimp sealed it, now it has integrity. You don't recognize that now it has oxygen because it had that temporary leak. So if you do headspace analysis and measure the oxygen content in the headspace, you would identify that it had leaked temporarily and it now has a oxygen content that's greater than the specification. This is also true for there's more of a like in Annex 1 requirements of confirming that you maintain the vacuum of your product. So some of these lyo products, they intentionally want to remain or keep a vacuum in there to help assist with the addition of diluents. But again, if you have a temporary defect, you may no longer have a vacuum. So you may have integrity if it's been crimp sealed, but you may no longer have the vacuum required to pull in the diluent from the syringe itself.
Thank you. Let's see, going back a little bit, actually the very first question, was about the practice of what to do for some of the testing limitation, initiation and completion from the pull date for time points less than 3 months and common practices there. And we talked a little bit. Emily is had to go, but she was talking about freezing things. And I was thinking back to my early experience. If it was that early on, sometimes we did hold samples in order to run them together. That got mixed um, approval from various regulatory people passing through on inspections. We did do it if there was close together, but we also stored things if we didn't have any data to show that it could be frozen. We would store it at controlled room temperature or laboratory temperature being monitored, and that always received an approval as we did that particular practice. But lots of times you just need to bite the bullet and do more of the testing, especially if the regulatory people come through it and don't like you holding samples. Walter, did you have any other experience on that from your career and what to do with those short periods?
Embarrassingly, our group, my former company did not have, they just had a 1 month initiation requirement across the board from time zero to 3 years. We were looking into alternatives, going to 2 weeks and implementing early pulls for later intervals and such. But we were never questioned on that, to be honest. We ourselves were a little uncomfortable at times, and especially in light of industry. But I'm glad we're revisiting this, just because I was hoping we'd hear more from more of you guys, since the question is asking for what is our common practice regarding that.
Yeah, I think we're going to see more and more of this question being raised with some of the biologics that can go out of spec faster once they're thawed and stored in a doctor's office. We'll probably also see a lot in the accelerated stability things like you see over in FreeThink Technologies using the short term by quite a bit. So it's going to come up more and more often as to what people are doing in the very short term. I know we had products that were going to go to very high temperatures. We had a request once from India to run some studies at 50 degrees C, which in my early career, we used to do that routinely, and then we dropped back to the 40 once all the guidances came out. But 50 degrees came back as an example of what they wanted, and we were doing weekly testing in order to find out more accurately what was happening at those higher temperature stages, because they represented excursions and things being out of their typical storage and protective containers to do that. So more and more people will be doing the short-term testing, and I think being responsible to test in the 1, 2, 3, or 4 week ranges. So opening that back up again, anybody else commenting on what they've been doing with their short-term testing, particularly at accelerated conditions?
Yeah, I could speak to that a little bit. It's going to be different than a traditional ICH study, but the way we run our studies, which are typically 3 weeks, is a pretty normal study. So we might have samples that are 1 day samples, 1 week, 2 week, 3 week. We actually kind of flip the way that the stressing is done, in that we don't set up all the samples on day zero. We actually stagger when things go into ovens such that everything comes out on the last day. So then when things are, before they go into the oven, they're kept at whatever storage condition is, refrigerated or ambient. But this allows us to pull everything and analyze it in one analytical run. So that greatly reduces variability of bringing up the method on different days by different analysts on different instruments, anything like that. Also allows us to randomize the order of samples. So if you have any kind of drift in the method from first injection to, in our case, 30 or 60 or 100th injection, we'll have controls, unstressed samples littered throughout the run, high stress samples at the beginning, low stress samples at the end, kind of a mix. So we kind of take advantage of that and flip the traditional approach of how the staging and pulling and testing is done.
Yeah, I brought up this topic because we're starting to get stability studies that have samples at 2 weeks time point and 1 month time points more often than what we've been seeing. The typical ICH time points are like 3, 6, 9. And we're trying to balance between the laboratory practice with the workflow and the auditor comments when they go through and asking, okay, if you pull a sample at 2 weeks, how long do you give the test? Do you give the lab to test those 2 week samples? If we say 30 days, then it's like hitting the next time point already when you're not even done testing the 2 weeks and your 1 month already comes out. So we're trying to figure out the best way to word it in the SOP, I guess. But currently we have 30 days for initiation, at least the initiation part of it. Like the sample has to be prepped or run on an instrument. We do have a guideline for starting at 2 weeks and completed by 30 days, but it's not a deviation if we don't do it within 2 weeks or complete it by the 30 days. We just have to initiate the test within 30 days. But obviously it's being questioned.
Yeah, I think that's very typical to have that question at shorter ranges. I had forgotten about the onset of testing aspect of that. Yes, typically if for accelerated studies, especially in the earlier parts of a protocol, it's not unusual at all to see a much abbreviated onset time if your analytical lab can handle it. So that's the key, is to be able to balance the resources that needed to do those initial analysis. And sometimes we even broke it down to a key analytical method that we needed to use, where some of the other ones who aren't seeing the degradation or the going south on us very quickly, we just focused in on the main assays, the ones that could change within a matter of a day or two, and very quickly get those done, and then put in our procedures that the other ones could follow in a reasonable amount of time. So I think that's typical that you'll see that, but just making sure the resources are there is the key, especially if you run into a rush season like you find at Christmas, or people going on vacations in the 4th of July timeframe of the year. It becomes very difficult to fulfill your promises on how fast you'll get those assays done. Okay, did we hit
Thank you.
that all right? You're welcome. We have about 3 minutes left, and I don't see additional questions here. We have some that we could ask, but I think it's a good time to talk about the future for these webinars and what's coming. So if you bring our program to a conclusion today, we'd like to invite you all to join us on Thursday, November 20th at 12 p.m. Eastern, when Parameter Generation and Control will sponsor a program covering vital details for recovering from stability program excursions, with both respect to equipment and the product impact evaluations, which is something that gets left in the dust many times. We're so focused on the chambers and getting them back up and the product back in there that a lot of times the regulatory authorities can start asking us questions about what happened to the samples. How can we prove that it didn't get thrown out of specs, that didn't shorten the shelf life, that we might be ignoring the long-term impact of those things. Do we need to shorten the shelf life or extend the period of time if our conditions were softened rather than hardened? So we'll address all those things and try to help out people in understanding what's going on with their studies when they have excursions. And on December 11th, Novatech International will be back with their final 2025 installation of stability management digitalization. So what do we do to make our studies more efficient and easier with less errors and less intensive resources? So all that will be coming up. Watch for details of those events in the monthly newsletter and in the events section of stabilityhub.com. And looking forward into the future, we have a number of new programs, new sponsors. We're looking forward to having even greater participation. So I'd like to thank our panelists for sharing their time and expertise with us today, and you, our audience, for bringing your questions and for attending and sticking with us. We hope to see you again real soon. And we thank you very much.
I know.